Okay, I have a few slides to finish here with principles of chromatography, and then I'll move on to HPLC. So we'll get started where we left off last time. Who can define peak resolution? When we say peak resolution, what do we mean by that? Simple. Yes, Jean? Say that again loudly. Okay, so resolution, Jean is saying, is related to capacity, selectivity, and efficiency of the column. Yes, but when I say peak resolution, what am I really meaning, Chrissy? Yeah, simply, it's just resoluting two components from each other or multiple components from each other, separating them. That's what we mean by that. Yes, it is impacted by all of the factors that Jean talked about. What is a front peak or solvent peak? When we say solvent front or a solvent peak, what does that mean? Yes, Michelle. Exactly. So what Michelle was saying is basically the solvent, when from the point of injection until it gets to the detector recognizes the presence of the solvent from the moment of the injection. So that's the solvent front. And it's actually, sometimes we call it also the void volume, because it's really nothing is coming out potentially unless the compound is not interacting with the column. It will come out with the void volume. So it's kind of like the volume of the column without anything else in there other than the solvent. Okay, what is the difference between retention time, volume, and retention volume and retention time? When I say retention volume, what do I mean? When I say retention time, what do I mean? They're not the same. Okay, so one is the amount of volume and the other one is the amount of time. For what? For a compound to come out. So, and they're not equal, obviously, because sometimes you have a flow rate of 1 ml per minute. So maybe 10 ml is equivalent to 10 minutes in this case, but flow rates are different. So that's why there are two different things. When we say retention volume is the amount of volume that it took for a component to come out, whereas the retention time is the amount of time it took for a component to come out. Okay, what is the relationship of peak width to resolution? Thank you for all of you that are responding via chat, but also, Anastasia, do you want to, yeah. Exactly. So the narrower the peak width, the better the resolution. So smaller, specifically the width, not necessarily smaller peaks. The width size, not size, but the width. The smaller the width, the better is the resolution. The sharper the peak, the better that two compounds that come close to each other, they will be separated. If the peak is broad, two compounds that would supposedly come out close to each other will just overlap in this case. So we really want sharp peaks. What is the first step in developing a separation between solutes of interest in your sample? What do you want to do first? Very simple. First thing, you have certain components. Yes, Dave. Yes, just look up literature to see if there are, yes, exactly. Those of you answering online too, look up literature to see what's out there. Somebody must have done this before. It doesn't work 100% each time. Even if a method is published, sometimes you just have to tweak it based on what you have in the lab. But somebody must have tackled it in the past. One thing I had added a question here when I was in the office, but it didn't seem that it saved. I closed before I saved. I just want to pick up where I left off. And I know that Jean answered this earlier. I had a question here saying, what are the functions that impact resolution? And Jean answered it when we asked about peak resolution. I just want to remind you that there are three functions: efficiency, selectivity, and capacity. So these three functions impact resolution. We spent quite a bit of time last time talking about efficiency of the column and the number of theoretical plates and such. So now I'm going to move on to selectivity. So the more selectivity you have, the better is your resolution. And you can control selectivity by changing or by selecting your stationary phase and also by selecting your mobile phase. So let me give you an example, which is actually it can help you answer one of the questions in the lab report for the HPLC lab report. So let's say I have two components that are very similar in their polarity. And they're different, but they're very similar, and they're like more organic phases than, so they like to associate with non-polar more than polar. Okay, so I have these two compounds, and they are interacting with the column. And when I increase the concentration of the acetonitrile, they both like acetonitrile, so they come out together in a way similar to these two peaks that are not separating in the bottom here. So I have this situation. I have a very good reverse phase column, and then I have these compounds that are interacting strongly with the column. When I add acetonitrile, they tend to come out very close together. So I want to enhance selectivity and differentiation between these two compounds. I say, okay, I'm going to replace acetonitrile by methanol. Instead of using acetonitrile, I'm going to use methanol. So why? How is methanol different than acetonitrile? Does anybody know? Yes, thank you, Jean. It's more polar. So methanol is more polar. Yes, some of you also answered. Sam answered more polar. That is correct. So now I enhance the selectivity where one of the compounds that are a little bit more hydrophobic than the other one will be retained longer on the column, and the other one that is less hydrophobic will go out with the methanol. So I can enhance separation, and really then if I want to reverse my arrow, so this situation can go to that situation where I have enhanced my selectivity. That's a perfect example how you really can control your mobile phase, in this example, to enhance selectivity between two compounds that are very similar in their characteristics. One thing to note here is selectivity carries more weight than efficiency. Why is that? If we look at the equation of the resolution here, so the number of theoretical plate, there is a square root of number of theoretical plate, and there's also a division by 4. So it's quadratically associated with resolution. However, selectivity is directly related to resolution. Okay, the last one is capacity. This has to do with how much functional groups you have available for your compound. Over time, you lose selectivity. Why? Because as you're running your system and then mobile phase is going through the column and the column is being overused, some of these functional groups are going to be shed off. So over usage, you're going to lose some of the functional groups. And by doing so, losing some of the functional groups, you have less interaction with the column. The components don't have as much functional groups to interact with. So they tend to leave early. So your retention time becomes shorter. So this compound here, A, instead of coming out, let's say in X minutes here, 10 minutes, let's say, it starts to come out at 8. So you have less time spent on the column. With that, if it's coming out sooner, then it's going to potentially overlap with another component that used to come out at that retention time. So I really reduce the capacity of the column. That way, over time, over usage, when my retention time becomes shorter. So also capacity plays an important role in a different way. And I'm going to talk about it when I talk about the different column, the thickness of the column, whether it is a capillary column or versus a packed column. If you have less stationary phase, potentially you have, or less amount of particulates that the compounds are going to interact with, you reach equilibrium faster and you have less diffusion into the stationary phase. So you can get sharper peak, but then you have less capacity, so you cannot inject as much sample. So your sample concentration should be really dilute when you work with low capacity surfaces. But we'll talk about that more when I start talking about the different columns and how they're packed. So when we do analysis, whether it is GC or HPLC, we always think, okay, what are we looking for? What's the ultimate goal? Is it a qualitative or quantitative? So the qualitative basically is detecting, identifying. I just want to know what's in there. That's detection. Or I want to know if it's there or not, presence or not. Do we have a certain component in there or we don't? So qualitative analysis. Quantitative is when we really want not only detect and identify, we need to quantitate. So in terms of qualitative analysis, it's detection and identification of a certain component. We do that when we have standards. So if I want to detect an identity of caffeine in my sample, I really have to run caffeine through the column to see the retention time and compare it to the retention time of the compound in my sample. So retention time is the first thing I would look at. Same with those of you that did the GC analysis, where you had to run a reference standard, and then you looked at the retention time to determine which frame is which in your sample. I gave this example of spiking in for the HPLC labs that we had. And I showed you the difference when you're running a sample through a detector that only gives you a peak at a particular retention time and you don't have any other information. You don't have a wave scan, you don't have any powerful detector. You're just getting a peak at a particular retention time. But then you have like 4 peaks are close to each other and you don't know, one is coming at 15.5 and one is coming at 16. You don't really know which is which. So when you have that problem, you can spike the sample. By spiking the sample, meaning you add to your sample a known amount of your standard, of that, like caffeine. If you're looking at caffeine, you add a known amount of caffeine, then you run the sample and see which peak got increased in height or in area. Then you would know, okay, so it's the 16 minutes, not the 15.5. We talked about photodiode array detector in lab. There are a couple of groups that haven't done that lab yet, but a photodiode array detector is a detector where it can give you a wave scan, measuring absorption over a range of wavelengths. So it can give you a little bit more about the identity of the sample, kind of a fingerprint of the sample. And I'll talk more about that when I cover HPLC in a minute. Detection using multiple detectors. You have components. Some of them absorb light, some of them don't absorb light. So you have this mixture and you want to identify components in this mixture. So you might say, okay, I'm going to have a UV detector, and in line with the UV detector, I'm going to have a refractive index detector. So then I can measure components that absorb light and components that don't absorb light and differentiate them. Peak ratio at 2 different wavelengths. If you have a UV detector that doesn't give you a scan, What you can do is you can measure absorption at one particular wavelength and then measure it again, measure absorption at a different wavelength. Let's say I'm going to measure at 250 and then I'm going to measure it at 275. And then I'll do the same for another component. They will have different ratio. Then I can identify them based on their different ratio. Separation using chromatographic, another chromatographic mode. That means 2D chromatography or 3D chromatography. And I'll give you an example at the end of the HPLC lecture on that, where oftentimes we need to do this, especially an example of that is in protein separations. So sometimes you have two proteins with the same molecular weight, but have different charge. So if I use size exclusion, I might not be able to separate these two. I might separate them from a third component, third protein that has a different molecular weight. But now I have separated one out of the three. If I have three, a mixture of three, two have the same molecular weight and one have a different molecular weight. So I separated one out of the three, and now I have two in one peak. I take that and separate using ion exchange, so I can separate both of them via ion exchange. Because protein is a perfect example where multiple proteins can have the same charge and multiple proteins can have the same size. So to get good separation, I would use multiple modes of separation to isolate all the components in the mixture. Mass spectrometry is a more powerful detector than UV detection or diffractive index detection. It gives me structural data. It can give me, like Gary is going to talk about MS Wednesday and Friday. It's not only going to give me molecular weight. Some powerful MSs can fragment and give you molecular weight of the fragment. So you can get structural identification. What different functional groups you have, you can piece it together and identify your compound. It's a very powerful tool, and we'll have a lab on it. The last thing I want to talk about is the quantitative analysis, which is oftentimes what we want. We want to quantitate components in foods. So we really not only want to identify and detect compounds, we really want to quantitate them. And without standards, it's really hard to quantitate. Sometimes you quantitate based on relative area. So you look at the different components and you determine the total areas, add the areas together and get the total area, and then you get the relative distribution. That's not really quantitative, that's semi-quantitative. You're getting the relative composition, relative to each other. But if you really, really want to quantitate in terms of a concentration, you need to have a standard curve. You need to have standards, and those standards, you need to prepare them at different concentrations. You need to get the area, yes. Couldn't you take the integral? And what do you mean by integral? There's so many different things that an integral means.
Instead of like estimating, couldn't you just like use calculus to find the area under the curve?
Yeah, well, you find the area under the curve, but then what do you do with it? The area under the curve, let me explain this, Aaron, a little bit. You can get the area under the curve all the time, but what does the area under the curve tell us? Every compound has a different response to the detector, okay? So two compounds might absorb at 280 nanometer, let's say, but their response, the area under the curve is different even if they have the same concentration in your sample. So, and that's actually in the GC lab report, you have question relative to that, is every component, every phane has a unique response to the FID detector. So even if you have them at the same concentration, the area under the curve might not be the same. So we cannot relate the area as itself to the concentration because it really is related to the particular response of that component to the detector. Good though, good that Aaron, you brought this up so I can explain this a little bit clearer. Okay, so now we get the area under the curve, and in order for us to convert it to a quantity, we really need to have our standards. We need to prepare them at different concentration. We need to run them and get the response at each concentration for that particular standard, and then we plot it. We plot it, and then we get a standard curve. But before I go into that, what do I plot? Do I plot the area under the curve, or do I plot the height of the peak? So the peak height can vary, and it varies based on how steep how sharp is your peak. If you have a great resolution, your peak might be very sharp and you have a really high height. If there's any difference in the efficiency of the column, your height might get smaller and your peak might widen, but the area would ultimately be the same. So it's better to use peak area for quantification rather than peak height. And in the past, people used to, when you didn't have the computer software, you kind of get a strip chart recorder, and it kind of record on a piece of paper, and then you can take that piece of paper and cut that triangle and weigh it. And that's how it used to be in the past. But now, of course, we graduated from that. We have softwares, we have the responses converted to electrical signal, and it gives us the peak, and then it calculates the area for us. Then we take this area, as I said, and then we plot it against the concentration, and we get a standard curve for a particular component. So we call this external standard. For quantification, you really need an external standard run at different concentration. Definitely. Now, there is something we call internal standard. The internal standard, the purpose of the internal standard is to correct for sample preparation and any errors in volume of injection. So what does that mean? So when I have an internal standard, I put it at the beginning of my sample analysis. I add it to my sample at the very beginning, and then I start my sample prep. So the internal standard is actually going through the same procedure as your components within the sample. So any errors that might cause a loss, underestimation, or overestimation of your different components will result in the same error to the standard, to the internal standard. How is that beneficial? Yes, we add the internal standard to our external standard mixes. So we have our external standard, different concentration. We add the same amount of internal standard to all of them. And I add the same amount of my internal standard to my samples. So I have the same amount of internal standard in everything. When I want to plot my standard curve, again, remember I need the external standards. I need them for quantification. So when I plot my standard curve, in this case, for every component, let's say I'm looking at glucose, fructose, lactose, 3 components in my sample. So instead of just plotting the absorbance versus the concentration, I have an internal standard. I'll plot absorbance of my unknown, whatever it is, glucose or fructose or sucrose, over the absorption of my internal standard. So I'm correcting for the internal standard. So I will have that. And then when I want to get the concentration of my unknown sample, again, in the y, so you have the equation of the line y equal ax plus b. So x is the concentration. So when I want to calculate x, I have the y, and the y in this case is the absorption of my component over the absorption of my internal standard. Okay, so I always have that ratio. I use the ratio to plot the curve. I use the ratio to calculate x. So I have my internal standard corrected for everywhere. So again, the internal standard is used to correct for sample error, sample preparation error, especially when you have sample preparation, same as the GC sample preparation, with a lot of steps and a lot of places where you can lose samples when you are determining FAME, or fatty acid methyl, or fatty acid methyl esters. So there are a lot of steps that gets you to that FAME, to the GC analysis. So if we have an internal standard, we can correct for these errors and have more accurate and accurate results, basically. Two things you have to remember about the internal standards, two main things. In order for it to qualify as internal standard, it has to have similar structure to your components of interest. It's very important. It has to have a similar structure so that it will interact a similar way that your components are interacting with the column. That's one thing. The other thing, it's important that it does not co-elute with any of your sample components. So in this case, for example, sugars and molasses, you have glucose, fructose, lactose, and sucrose. So in molasses, you don't have lactose, right? Lactose is sugar in milk. But you have glucose, fructose, and sucrose. So you can add lactose as an internal standard. It will still come out in your chromatogram. It will not co-elute with anything. And it will come within the analysis time, not something that will come out 2 hours later. Also, you want to make sure your analysis time remains reasonable. So within the same analysis time and does not co-elute and has similar structure. Yes, Rachel, is there a question? Yeah, first asked, but doesn't the internal reduce chances? Exactly, that's what it means. Using the internal standard. Is your volume on? No, they can't hear you. But everybody can see the question. Using the internal standard account for human error. That's exactly why we are using internal standard, to account for human error that can happen during sample preparation. So you account for that. Can I answer your question? Oh, that's okay. All right. Any questions about external versus internal? One thing I want you to remember, you need external for quantification, internal to correct for errors. All right. So there are these two YouTube videos that are really nice to watch on your own time. I will make them available on Canvas, but there's one for HPLC and one for GC. All right. So moving on to the HPLC. These are more recap questions for you to study from. Okay. So HPLC, it used to be called high pressure liquid chromatography. So we have the low pressure liquid chromatography we talked about in the past couple of lectures, and then we have the high pressure liquid chromatography, and it used to be referred to as high pressure because we pack the column under really high pressure. So your material that is in the stainless steel column is really packed under high pressure. But the performance was improved over time, and the packing technology improved over time now that you don't generate as much back pressure as we used to in the past. With that, the nomenclature for the P changed from pressure to performance. So now it's high performance liquid chromatography. This is basically the different beverages that you would prepare in the lab, where you dilute and then run them. One of our former TAs did that, and I thought that was a really nice illustration. So if we want to compare high performance liquid chromatography to traditional low pressure liquid chromatography, we have enhanced the speed. So the analysis time is much shorter, and the resolution is better. Why? Because the material, the stationary phase, is packed. There's not a lot of void within the particles. When you don't have a lot of void, your particles don't have much places to go other than between the stationary phase and the mobile phase. That it will not get lost in some of void. So you have less diffusion. Basically, you have sharper peaks, and you have and the compound leaves the column in a shorter time rather than just going in and getting lost in the interspatial void. So that's why we have better speed and better resolution. Reusable columns. So we do purchase reusable columns. So the column that you've seen in the lab or you still will see on Wednesday is a stainless steel column that is packed under like solid pressure, high pressure. So it's packed in a way that makes it, you can reuse it multiple times. It won't, whereas low pressure chromatography, sometimes we have to empty the column and then repack it to ensure that the material is still, doesn't have a lot of void and it did not get displaced or disjointed. So it is they are reusable, but they're very expensive. So depending on the stationary phase in the column, this price can range from anywhere between $600 to $2,000. Average is around $800 to $900. It's kind of common HPLC columns. They are reusable, but that doesn't mean that they don't die. They will die out. First of all, if you don't maintain them, cleanse them, equilibrate them, store them in the right buffer or right solvent, they will die out on you. They can dry out, they can get dried and break internally, or they can just lose functional groups if you store them in the wrong phase or mobile phase or solvent. So they can be used for a long time, but at one point they're going to lose efficiency by just simply being old and losing functional groups. And then you get easy sample recovery by that because you have good separation and resolution. You can collect purer peaks. Usually in low pressure liquid chromatography, your peaks are not sharp and you kind of have overlap and the peaks might not be pure. And here's the link to the YouTube video that you can watch. It's like 10-15 minutes long, but it's really informative. So these are the main components of the HPLC. You have the reservoir for your mobile phase. Sometimes you have 2, 3, or 4 reservoirs, depending on how you want to change the mobile phase during the run. You have, commonly, we have a pump or 2 pumps. Sometimes you have 1 pump and then you mix under low pressure your components and pump them into the system. Sometimes you have a pump for each mobile phase, and then you mix under pressure. After the pump, what the pump does, it's pumping the mobile phase at a constant rate under a constant pressure. And it goes from the pump to the injector where the sample is injected. And when the sample is injected, it gets carried by the mobile phase into the column, and the column contains your stationary phase, and that's where the interaction of the different components happen with the stationary phase, and they get separated. And hopefully, you would hope that they get separated, and as they separate and leave the column, they get to the detector. They get detected depending on whatever signal. And from there, the signal gets converted to electrical signal, and you get a chromatogram, which you can analyze. After the detection, the sample and the mobile phase continue their way into the waste, or sometimes you collect the sample to do other types of analysis. Maybe take it to NMS, or take it to NMR, or do an enzyme assay on it, or something. You want to collect your fractions, or even do a taste panel on your components, taste component. That's one of the studies we're doing, using LC, separating taste components in cheese, and then having the sensory panel taste the different components as they come out from the column. So in order to collect the different components, we have fraction collection. a compartment where it would be timed, like after every 5 minutes, it collects a certain amount of sample and moving on to the next. Who's up there? Okay, so we can link that if we need to collect samples. So the pump allows for constant pressure and constant volume. That means it controls our flow rate. For example, in the caffeine lab, we had a flow rate that was fixed at 1 ml per minute. And flow rate is important to get the desired resolution within the analysis time that is reasonable. We can have low pressure mixing or high pressure mixing in case of our gradient elution when we're using multiple solvents. So with low pressure mixing, that means I have one pump and let's say 2 solvents. So the 2 solvents get mixed before they enter the pump, that we call low pressure mixing, and then they get pumped under pressure into the system, into the injector and so on. The high pressure mixing is when I have 2 solvent, 2 pumps, or 3 solvent, 3 pumps. That means each solvent is pumped under pressure separately and then mixed in a mixing compartment under pressure already. High pressure mixing usually have a much cleaner or smoother baseline where you achieve better mixing between the 2 solvents. So my preference is always for high pressure mixing. So injector, you have an auto injector or a manual injector, and some of you already seen those 2 in the lab. With an auto injector, you place your samples in a tray, and you can, sometimes a tray takes 50 samples or 100 samples depending on the size of your equipment. And then you can program your software to have a sequence where samples are injected in sequence, and you can just put your samples in and leave and come the next day and you'll have everything analyzed. However, if you have multiple samples and you know components might degrade, you would have a compartment, we call it a cooler, where your tray is cooled down to refrigerated temperature, like 4 degrees or something like that. So this kind of analysis, usually if we have components that would degrade over time, you want to make sure they don't degrade during your run so that you don't have the first few samples running nicely and have good data and the last few samples are erroneous due to degradation. So you would cool them using a cooler. So the auto injector has its own needle that would automatically go to the designated spot. The needle goes through the septa of the screw cap of your vial and then withdraw a sample and inject it into a sample loop. So you have a compartment here that you attach your sample loop. That's where your injected sample goes through. Sample loop can be 20 microliter, 50 microliter, 100 microliter, depending on how much you're injecting on the cone. So actually, you can change the sample loop size. And what happens is you clean the sample loop. So you inject the sample or inject cleaning solvent, and it goes directly to the waste. Or when you inject your sample, the amount that is designated to go into the column, the route opens between the sample and the injector here, where the mobile phase actually go into the sample loop, carry your 20 microliter sample or whatever, 50 microliter, whatever you programmed it, and takes it with it to the column. When you're done injecting, the valves are closed, and then you will have your mobile phase just circulating without going through that sample loop. So it will bypass the sample loop. The manual injector has the same principle. Instead, you would inject with a needle and a syringe. So you have your syringe, you have a specific needle, and you manually withdraw the volume for injection, and you manually inject. Manually clean the loop, manually inject. Columns. Columns can come in different sizes. So you would have a column, the length would be different, the width would be different. So you can have long columns, short columns, thick columns, narrow columns. So it depends on how much sample you want to inject and separate and how much resolution you want to get. Longer columns, narrower columns give you better resolution. So with longer columns, you have more time for the components to separate, and narrower columns will have less stationary phase, less diffusion, and samples will equilibrate faster and leave the column faster. You have shorter analysis time, and you have better resolution. 3 columns. So before you have in the flow, you have your, after your injector, you have your column. So your mobile phase is going from the injector to the column. But before it gets to the column, oftentimes you have 3 columns. We call them mostly guard columns. So they're guarding the column. So you have them at the beginning before the sample reaches the column, and it's basically capturing any impurities that might interact strongly with the column and block the functional groups on the column. So your sample goes in, well, you filter your sample, right? But you still have some impurities in there that might interact strongly with your column. So you want to prevent and extend the efficiency of your column and the life of the column by using a guard column. The guard column basically kind of acts like a filter, just filters out impurities. It gets full at one point and you have to replace it. And it's packed with the same material as your columns. Whatever your column stationary phase is, your guard column would be of the same components so that it can capture whatever impurities would otherwise be trapped on your column. The different types of HPLC columns, you have the microbore, which is very common, one of the most common, with an internal diameter of 0.5 to 2.5 millimeters. That's a very, very common HPLC column. Other columns that actually go with ultra HPLC is the micro columns or the capillary columns. So they have a very, very narrow diameter. So the difference between micro column and capillary column, they're both small or have narrow diameter, but the capillary column have a it's a tubular column where it's hollow from the inside. It's hollow from the inside, and then you have a stationary phase, a thin layer of the stationary phase on the sides. So similar to GC columns, all GC columns are now capillary, not packed columns. So your sample goes through with the mobile phase, and it has a very, very thin layer of stationary phase to interact with. So you don't have a lot of diffusion. So the sample, the components have only like surface kind of functional groups on the surface. They interact and they go in and out into the mobile phase, and they reach equilibrium very fast. So you really have very sharp peaks with the capillary column. And they actually, with a capillary column used with ultra HPLC, you only can inject very, very small volume, very low and diluted sample. Otherwise, you overload the column. You don't have much capacity there. So you can analyze really dilute sample, but then you would have very sharp peaks. So you get good signal from your detector. So you have good, powerful identification detection. Powerful detection, because you do have very sharp and with very high height. So in general, packed microbore and microcolumn, both, they are packed. That means that there is no hollow empty hollow. They're all packed, and they're packed very well so that you have minimum void, so you don't have much void between the different particles, and the particle size is small so that you don't have a lot of void. So you can pack them really well, and that can enhance resolution, obviously. So the advantage of using smaller diameter columns: increased resolution, increased peak height, causes enhanced detection and sensitivity, and reduced equilibrium times. I see D. Chrissy? I think I agree with you, Chrissy. It's all of the above. So you do increase resolution, you increase peak height because you have very, very sharp peaks. So your height goes high, you have minimum diffusion, so you don't have broadening of the peak. And that reduced equilibrium time because you don't have diffusion, so the molecule doesn't really go in and get diffused or get lost between the spaces. It just comes out really quickly. So you reach equilibrium between the stationary phase and the mobile phase quickly, and faster equilibrium means better resolution. So they're all related, so all of the above is correct. So the packing material of the column, what we usually use is a packing material that can withstand pressure and has good durability, withstand pressure, withstand changes in temperature, withstand changes in pH. So it's really durable. It should have well-defined particle size and small particle size, and hopefully not too porous or doesn't have large pores. Because you have the particle size that is important, how big the particle is, small versus big. The smaller they are, the less spaces you're going to have in between. If they're very large, you have larger spaces. And not only that, if they're porous, so the size of the pores inside it matters. So the smaller the pore size, the better it is, because if you have larger pore size, you have, again, your components can go into the pores and get lost there or detained. So the packing material should, like I said, should withstand sufficient mechanical strength, that is, withstand high pressure, and good chemical stability that might also withstand not only pH changes, but salt changes sometimes when you're running buffer through your system. So we have different types of packing material or your stationary phase. Sometimes your packing material is silica, but bonded silica. So whatever is bonded to the silica, for example, in this case, an organochlorosilane with the R3 group here, this R group can be derivatized to be a nonpolar group, a polar group, ionic group. So it defines what your stationary phase is. So that we call a bonded phase. So the silica is bonded to your actual functional group. Sometimes you have an inert core that it's non-porous, so your sample, your components don't go through it, it's not interacting with it, and you have a thin surface coating or layer. This kind of setup, again, is similar to the capillary tube setup, where you have a very thin stationary phase, you have less chance for diffusion and going in and being detained, and you have better resolution and sharper peaks. And the functional groups can be any functional group, whether it's ion, exchange functional groups, or nonpolar or polar. So we have the bonded phase, the inert core, and then we have stationary packing or packing material that is polymer. So that polymer can be lightly polymerized, like in the micropores, or more polymerized, heavily polymerized. So we have the lightly polymerized and the heavily polymerized here. In this case, when it's lightly polymerized, then you have larger spaces. So this is mostly for molecules of larger molecular weight. So if you have molecules of larger molecular weight, you want to make sure they're able to move through your stationary phase and interact with the functional group. So you really need to allow them that space. The other one is for smaller molecules where you have this highly interlinked matrix that have smaller spaces in between, and that's for smaller molecules. These two setups are used quite a bit in ion exchange chromatography and for separating components with charge. This is a very common form of stationary phase. And again, depending on the size, we are separating whether larger size versus smaller size components. Ovens with, oh, I went over. Okay, I can stop here and finish on Wednesday.